I will describe the Cellosaurus in the context of rare disease research efforts. And if I want first to start with cell line in general, I mean, this will be obvious to a lot of you, but cell lines are of course used by labs in academia and industry all over the world. I mean, some are acquired by researchers from cell collection, like ATCC, Coriell, for example, a lot of genetic disease, DSMZ, DCACC, and of course many others are transferred between labs, which sends them, I mean, or shares them. And there's a lot of different variety of cell line. As this I will come back in the next slide when I will speak about cell line which are useful for rare disease, but there is quite a lot of varieties of different type of cell lines. And in terms of resource, of bioinformatics resource, are cell line catalogs from the different organizations which distribute cell lines. So ontology like Branda tissue ontology or cell line ontology or EFO, which describes cell type and sometimes cell line, and many specialized databases. But until the Cellosaurus was established, I mean, already 9 years ago, there was no single resource where all this information was made available. So about cell line for rare disease research, I mean, basically you could say that 5 categories of cell line are widely used in the context of rare disease research. First, all of the finite cell lines, which are often fibroblasts obtained from skin biopsies of patients with genetic disease. So quite often those are established at the time of diagnosis. And while those are finite resource, I mean, they're not immortalized cell line, they can be banked, they can be distributed, and they're, I mean, generally distributed by different entities like Coriell or some biobanks. Then you have a big category of EBV transformed lymphoblastoid cell lines. So those are obtained from the peripheral blood of patients and immortalized using EBV. So there's quite a lot of those cell lines which are used in a context of rare disease research. But of course, I mean, in the last 10 years, what has been exploded is the use of iPSC-induced pluripotent stem cells, which are generally established from the finite cell lines, the skin fibroblast or the EBV, or just peripheral blood cells from the patients, and which are, I mean, basically transformed to be iPSCs. So there's also some pre-implementation genetic diagnosis derived human embryonic stem cell lines. And finally, when we speak of rare disease, we need to speak also of rare cancers. And then, of course, there is also cancer cell line originating from patients suffering from those rare cancer. So those are the 5, I would say, different categories of cell line which are quite important in the context of rare disease research. Now, first one summary slide on Cellosaurus, and we'll go back into more detail into it, and then we'll zoom in into things which are more important for rare disease. So it's a knowledge resource on cell line. The release, which will come out in about 2 weeks, will have about 128,000 cell lines. And its scope is basically immortalized, naturally immortal and finite cell line, but not primary cells. And it covers vertebrate and invertebrate cell line, but not plant cell line. But a majority of those cell lines are human cell line. 90% of all the cell lines in Cellosaurus are human cell lines. So there is about 50 different types of information items, many literature reference and cross-reference. We'll come back to it. It's available by web access on Expazi. We'll also see that. And you can download it in 3 different formats, either in text mode, OBO, or XML by FTP. And it's available by the CC BY license. It's part of the Resource Identification Initiative and the International Cell Line Notification Committee. I won't discuss this in detail because that's outside the scope of rare disease. And one thing which is inside the scope of rare disease is the tight collaboration between Cellosaurus and the Human Pluripotent Stem Cell Registry, which caters for iPSCs and which has many cell lines, iPSCs for rare disease. Since January this year, it became an Elixir core data resource. Now, what can you find in Cellosaurus entries? This I will go quickly through it because it's not, I don't want to spend a lot of time in it, but we'll come back after to the important parts which are mundane to rare disease. So of course, you need a name for a cell line. You have often synonyms because people are often, either abbreviating it in different ways or renaming them. So there's a lot of synonyms for cell lines. You have also misspellings, which are important to capture because sometimes cell lines get misspelled, and the misspelling is often sometimes even more common than the right name. We want to capture the category of cell line. I just told you about those transformed cell lines, stem cells, and so on. Where it came from, the sex of the donor, the age of sampling of the donor, which species. If it's not human, the breed and subspecies of the animal where it came from, the anatomical origins, the organ, tissue, or cell type, and some information which are important from cell line, like population doubling time. And here I will go quickly because it's less mundane to rare disease like macrosatellite instability, karyotypic information, which can be useful, of course, for rare disease. And here, two very big, important topics, which I would come back to, is annotation of the disease in which a cell line is involved, using both NCI Thesaurus and Ordo from Orphanet, and the description of important sequence variation compared to wild-type genome of the species. And for human, of course, it's compared to the human genome assembly, and with cross-reference if there are those variations exist into ClinVar. Okay, for when you have gene which have been transfected, knocked out, edited, and there is this information about the gene with cross-reference to database, resistance to chemical compound. I would go quickly to this part because this is not really so important for, rare disease, but HLA typing, genome ancestry, for hybridoma as a monoclonal antibody target and a produced monoclonal antibody isotype. If it's registered in an official list, if it has been discontinued from a catalog, known contamination, misidentification cell line, STR markers, which are very important for cancer cell line because of the big problem of cell line contamination, which is less a problem for rare disease, except for rare cancers, because for most of the rare disease cell line, people are using cell line which are not so aggressively duplicating and overtaking, other samples. And so cell line contamination is not so much an issue for rare disease. Okay, and finally, but not least important, reference to publication and article, book chapters, and patent and thesis, and cross-reference to cell collection, cell resource, and so on. Quite a lot of cross-reference. This is just, a graph with some of the 93 now database which are cross-referenced, from the Cellosaurus. Now, so yeah, or before going to rare disease, the curation strategy, it's basically four different ways of getting information, either extracting data from cell line collection, which is not easily automated because those cell line collection product page are completely, maintained in different formats. Sometimes they're just HTML page and there is no database behind. Extracting data from external bioinformatic resource, which gather from specific data relevant to cell line. And the biggest source of information is manual curation from publication, about 21,000 publications so far. And what is increasing nicely is submission and personal communication, which is something quite useful when people submit information which can be added to Cellosaurus. Just a statistic of the current release. As I said, there's going to be a new release in 2 weeks with 128,000 cell line, but this release, which is currently available, is the release of January with 123,000 cell lines, 84,000 synonyms, about 120,000 reference to about 20,000 publication, a lot of cross-reference, web links, and a lot of cell lines with STR profile for authentication. Now, what is the relevance of the Cellosaurus for rare disease? Well, it's the only resource that centralizes and standardizes information on cell lines which are useful for rare disease. Now, if you take all of the cell lines in Cellosaurus, about 33,000 of the 128,000 are cell lines which are from patients suffering from a rare disease. So either a rare genetic disease or rare cancer. And currently it's about 1,200 different rare diseases. And those cell lines are from a lot of different organizations, biobanks, companies, and from a multitude of individual labs. And what's important in this context is to provide information on disease and on sequence variation. So how do we annotate disease? I said it before, we use two medical ontology to annotate disease affecting the donor of a cell line. So NCI Thesaurus for all human and non-human disease, because you have to remember that we have not only human cell line, but also mouse cell line, dog cell lines, and that you need to have an ontology which caters also for those diseases. And so NCI Thesaurus, to my knowledge, is the only disease ontology which contains both terms for human and non-human, species. But for, recently in March, last year, so exactly 1 year ago, we started providing and linking to ORPHANET Ordo. So for all human rare disease, we mapped to ORPHANET Ordo and gives the ORPHANET identifier for the rare disease. So currently 58% of all Cellosaurus entries are annotated with at least one disease terms, but that means about 2,100 NCI Thesaurus terms and about 1,200 Ordo terms. Wireless Ordo terms and NCI Thesaurus are more or less already explained before, because NCI also caters for non-human disease, and it also caters for non-rare cancer. So we have a lot of terms for cancers which are not out of the scope of rare disease. But you see that there is already 1,200 Ordo terms which are in use in Cellosaurus. Just about NCIs, they have created a value set which they call the Cellosaurus disease terminology. It's a bit misleading because we don't want to create our own disease terminology, but what they call value set are just subset used by resource. So they just basically collate all of the terms which are used in Cellosaurus and create it as one file for people which want to download or look at all the terms which are used in Cellosaurus. But again, it's not a specific terminology, it's just the set of terms from NCI Thesaurus which are used in Cellosaurus. Now here is the important part on sequence variation, because if we speak about a rare disease, it's important to capture those sequence variation. So we try to capture relevant sequence variation. So genetic disease causing mutation, for cancer, important oncogenic somatic mutation, gene deletion or amplification, and gene fusion. So those are described using the HGVS nomenclature, so this is not really a surprise. And when a variation exists in ClinVar, about 50% of our variation of human cell line, then we link back to the relevant ClinVar entry. Now here you got a screenshot of an entry from a current release with mutation. As I will describe in the next slide, this format, which is quite textual, here you see homozygous for ACVR2A and DB-SNP, or here ClinVar, cross-reference, is being changed for the next release, and the data is going to be completely structured in a different way. So what do I mean by this? So the next release will be the first to have completely structured sequence information. So we have 4 categories of sequence variation: gene amplification, gene deletion, gene fusion, and mutation. And for each of them, all the sequence variation are in a format which is structured with the AGNC, accession if it's a human, gene, the zygosity, and so on. So here you have an example for gene amplification and gene deletion, a gene fusion, and of course, the most prevalent mutation for genetic disease are single position, I mean, mutation or deletion or insertion, of course, but I mean, basically in a format HEVS with the clean var information. So this will be finished for the next release in 2 weeks, so that all of those sequence variation are now completely parsable and useful for, I mean, bioinformatic purpose in terms of making use of them. Of course, to read them as a human being, so I come back to here. This type of format is okay to read as a user, but if you want to parse it, the next format is going to be much more useful. We also, I mean, include inside the annotations engineer sequence variation, because more and more people are using CRISPR and Cas9, introducing mutation, for example, in pluripotent stem cells or in embryonic stem cells, to provide cell lines which, I mean, have mutation which are found in patients suffering from rare disease. Here you have, I mean, an example of a mutation introduced on preselenine 1, I mean, for the familial Alzheimer's disease, where they have edited a position and homozygously added this mutation using CRISPR-Cas9. In some case, a mutation in a disease cell line is corrected to obtain, like this, both the mutated and non-mutated cell line for the same genetic background. And so this is also indicated by saying corrected or edited. You see edited for adding a mutation and corrected for the correction of a mutation. So some stats about the sequence variation. 12% of all Cellosaurus entry contain at least one sequence variation, but for RD cell line, this percentage rises to 31%. So a third of all the cell lines useful for rare disease studies have an annotated sequence variation. And in total, inside Cellosaurus, we describe 6,500 different variations that affect over 1,200 genes. But if you look at those which are the human genes inside rare disease cell lines, so that's about 850. So we now have sequence annotation on mutation touching about 850 genes in the Cellosaurus in the context of rare disease. Okay, just to explain to people how you can get the Cellosaurus, I said already you can get it by the website, and we'll see that in a few minutes. But you can also download it by FTP, free format, as I said before, at this address. And there is about 4 releases per year. And as I said at the beginning, it's distributed under Creative Commons 4.0, I mean, license. I mean, what I can do now is, in fact, skip for a while inside. Do you still see my screen? Because I don't have any feedback back visually, but is it still correct, Yanis?
Yes.
Okay, so you have basically, I mean, from the Cellosaurus homepage, you have a description. You can basically you have a different number of options, but the most, I mean, the option people use most often, of course, is to search for a cell line or for other information. And I will give, and then you get a cell line entry or a list of cell line. If I just type, for example, something like, I will do this, I will show you again how better to do this than just by typing a term. But here I typed a term like cystic fibrosis, but I will show you that it's much better to do it differently than by just typing a text like this. But you see the principle, you type something and you get a number of cell line, and then you can go and look at those cell lines. Here I selected 2 examples already. One is an iPSC from a group in, I think, in Spain, where you have the mutation, 2 mutation, heterologous mutation in the SGSH, I mean, gene which causes Sanfilippo syndrome with a link to orphanet or to NCI-Thesaurus. And if I go down, you have the reference. You also have, of course, the links to the HPS resource from Germany. And if I look at another example of cell line for iPSC, here I have a cell line from Coriell, so Coriell resource, which has a lot of EBV transform cell line. You see here, it's a line modified using Epstein-Barr virus. And this is a cystic fibrosis cell line, again, heterozygous with 2 mutations. Again, the link to orphanet or to the NCI thesaurus. Well, I didn't say before, but of course you have the sex of the cell, the age at sampling, and the reference. And again, the links to different resources. Here it's to Coriell. Now, if I go back to the presentation, I skipped those 2 slides because I just add them in case I could not do this online. If you do text search, and you saw me doing a text search, you access currently an index, which is basically a full text index of all the world in Cellosaurus. So you can, of course, search for a cell line name, but you can search for a catalog number in cell line collection, a disease name, a species, a reference, authors, title, everything you can think of, which is in the entry. And this search bar is available at the top of every Cellosaurus entry. But what you need to do is try to do it in the smartest way possible, meaning that instead of using a string like cystic fibrosis, which is fine, you get results, but you will get everything which have an entry where the word cystic fibrosis is mentioned. And it could be that the paper title as cystic fibrosis, but the cell line itself has nothing to do. So here you see 302 hits for cystic fibrosis, but in fact, about 20% of those 302 cell lines are not from patients suffering from cystic fibrosis. As I said, it could be elsewhere in the entry. So how do I get this line which is only for cystic fibrosis? Well, you need to use the orphanet ID. If you do this, then you will get only those lines which are from patients suffering from the disease. So whenever it's possible, don't use a term, a string for the textual string to look for disease, but use an orphanet ID to search for. You can do this also for, variation. You can use an HGVS nomenclature that will work, or you could use a ClinVar variant, and this will work also. Okay, HGVS nomenclature is quite precise. So here, using this, either phenylalanine 508 del or the ClinVar number will normally give me the same results. Let's try to do this. If I do, P508 del, let's get one of those mutations and use the ClinVar number. So let's see, 167 hits. I didn't test this before, so if it's not giving it right, then I got somewhere a small problem. You see, well, oops, good. 157 also for the ClinVar. So there was no demo effect here, it worked. But basically, using also the description of the variation would work also. Except, of course, that the variation could exist in 2 different genes. Here, phenylalanine 500 del is unambiguous in a way, but you could imagine for another variant that you would find the same HGVS in different genes. So why not use the ClinVar number instead? Now, one thing I didn't say, and you would wonder why I speak about that, is we populated Wikidata with Cellosaurus data. Now, this was a project we started 3 years ago, where for each cell line in Cellosaurus, a concept of an equivalent concept was added into Wikidata. And this was done by master's student Lelia Teborn, and now it's updated by somebody called Tiago Lubiana in Brazil. And it was an interesting community exercise, but it's also useful in context of rare disease. And I will show you why. Well, first I'm showing you an example of an entry, of a Wikidata entry for, in fact, a cell line of a patient suffering from Xeroderma pigmentosum, with the information of that cell line, which is less than what there is in Cellosaurus, but it has information on the disease, which is inside with the terminology used inside the medical condition, which is used inside, Wikidata. Now, what does this allow us to do? Wikidata has a Sparkle query service, which allows you to do very, precise query, which you cannot do with text search. And here you have one, which is here searching for all cell lines which are associated with disease that have an order identifier and which are children of the Wikidata concept carbohydrate metabolism disorder. So instead of looking for one disease, I'm looking for a category of disease and all of the cell line involved in this category of disease. And if I do this and go to Wikidata, it will basically retrieve me 259 cell lines, which have different disease label and which are basically linked to orphanet IDs. And this currently you cannot do inside the Cellosaurus, but I will in a few minutes come back to it because we want to allow you to do this from the platform. But currently from Wikidata, you can do this on the Cellosaurus data. As I said, you can download by FTP the data inside the Cellosaurus. And you can also download a specific entry. Let's do this, in fact, here. If I have an entry, you see text version, you just click on this and you have just the record of the text version of the cell line and of the reference which appear. And you can get this, basically do this as an API. You can just, load any entry by having its accession number.txt like this. So that's a useful way of getting an entry in text mode instead of downloading the full set of all the entries into Cellosaurus. This is what I'm describing here with basically the URL in the form of Cellosaurus accession number.txt. And you have what I just showed you here. It's a different entry, but so you see the principle. So here I go back to what we want to, what we would like to add in Cellosaurus, which could be useful for RD, rare disease research. One of the things we would like to is to complete the scope of our cell line by capturing information for biobanks that are not yet represented in Cellosaurus. And here I have a gripe against a lot of biobanks. It's quite challenging. We have asked a few biobanks to give us just a list of the cell line with minimal information. The disease, the name, not the name, but the name of the disease or the order number, the sex of the patient, the age at sampling, and the type of cell line. And of course, the name of the cell line, and if possible, a link going back to the biobank. And most of them do not want to share this information. I don't know why. They think that it's either detrimental to their mission or no use whatsoever to the community. So, of course, resource like Coriell, there is no issue because Coriell is distributing cell line everywhere in the world and has a huge catalog which is online. But if you go to individual cell line, it's basically, either you don't get an answer when you get your email to ask them for this data, or you get two people asking you, why do you want this data? Are you going to order the cell line? If I tell them no, it's to put them in Cellosaurus, they say we're not interested. So if there are people here listening to this seminar which are in charge of biobanks or which have good contacts with biobanks, please try to do something. We need to have a full list of all the cell lines which are available for rare disease. And if this is hidden behind, catalogs which are not available anywhere and nobody can really make a list of those, then it's not really useful. So this is a big gripe, and we would like to add those cell lines to Cellosaurus. Now, one thing which can be added to Cellosaurus, which doesn't exist now, is there's a lot of cell lines which arise from donors that belong to a family of a patient suffering from rare disease, but which are not themselves suffering from the disease. For example, patients, heterozygous patient of a recessive disease. So in this case, we do not link to the disease because the patient, those donors are not suffering from the disease. So what we want to do is structure more information on rare disease in Cellosaurus so that we can basically, also add those familial relationships and map the disease, but by telling, users, this is a cell line which is, has this mutation, but which is basically not from a patient directly, from a donor directly suffering, but from the mother or father of patient X and Y. This type of information, for example, is available in Corial and in some resource, and we need to basically also do that in Cellosaurus. The second type of things we should add is, in fact, to do some software development tasks which are not going to be useful only for rare disease, but for everyone. We want to develop an RDF version of Cellosaurus with a Sparkle endpoint. So this allows advanced search and federated queries. You saw the type of search we can make in Wikidata if there was, when there will be, I should say more, when there will be a Sparkle endpoint for Cellosaurus, then those type of queries can be done from inside the Cellosaurus and from across different resources dealing with rare disease information. And we need an API for query and download of the information in Cellosaurus. So all this should allow us to develop a type of specialized portal inside Cellosaurus, which caters for RD researcher, where they can query what cell line are available for a particular disease or group of disease and scope of mutation available in those disease, for this cell line, sorry. Now, I will end now to basically be able to answer questions. I want to thank first, a number of people. Elizabeth Gastiger from Swissprot Group has implemented Cellosaurus on the Expazi server. Thibaut Robin was a PhD student which finished his PhD early last year, and he has developed a tool which I didn't describe here, but which is a tool which allows to look for similarity to STR profile for cell lines. This is useful in terms of authentication of cell line. Alain Gauteau, which has developed a parser to generate the OBO and XML version. I spoke already of Lelia and Thiago, which have developed the Wikidata Cellosaurus bot. And Amanda Kempis-Davis, which is the Secretary General of ICLAC, which has so many useful suggestions. And of course, a lot of individual scientists which are answering emails when I ask them for questions about their cell line, or which are submitting cell line information. And you can always send me emails directly on Cellosaurus email address for question, update, submission, and so on. You can click on the contact page and directly send fill the form and send an email from there. You can follow on Twitter what is new on Cellosaurus and news about cell line in general at the handle Cellosaurus.